Okay, so it's a pleasure to be here and to see everybody. Oh, great. So started off already. So I'm going to tell you a little bit about some of the work we've been doing in terms of engineering biomaterials for different kinds of applications in healthcare. I actually started off not as an engineer, but as a pharmacist, and then I did a PhD in single molecule biophysics. And now I work in an engineering department, and I'm a fellow of the Royal Academy of Engineering. So it's a real pleasure to be able to come and chat to you at this event. So I've pressed this thing. I don't know if it's, do I need to press harder? There we go. So I want to start thinking a little bit about how people have used biomaterials for healing the body for a long time. And what you can see in this picture here, this is a picture of a beautiful nacre shell. And this actually used to be used by the Mayans when they were missing a tooth. They would take this and they would put it into the jawbone. And because of the nanoscale and microscale structure of this material and the fact it's slightly mineralized, it would actually integrate into the bone within the jaw. And they would use this as artificial teeth. And over the years, people have used many different kinds of materials, natural materials, to help with repairing the body. People have also, in particular medics actually, have come across really interesting ways of using materials. And I love this example here, because this is an example from World War II. Sir Harold Ridley, who was an ophthalmologist, used to look at pilots that were coming back from their Spitfire missions in these airplanes. And what he noticed was that a lot of these pilots were coming back, and they'd have little shards of material in their eye, and it turned out to be from the plastic of the Spitfire canopy. But what was very interesting to him is that even though these little bits of material were in their eye, they weren't causing any irritation at all. So it was a very inert material. And he did some investigations. He found out it was indeed from the polymer that was making up part of these planes. And he had some of this polymer material made, and he ended up developing the first intraocular lenses from this material, which is called PMMA. And these are now used in millions of people all around the world. And it was really starting from that observation that you could have a material that would be inert and could provide this function within the body for medical use, that a lot of those initial investigations started out. And things have changed quite a lot. So from those initial observations of these inert materials, what we're doing a lot now within the field of regenerative medicine is actually developing materials that specifically interact within the body and are not inert at all. They actually really help in a tissue regeneration process. And this is very important if we think in terms of making better medicines, because you're all obviously very aware that we have an increasingly aging population. This is just showing some of the data from the United Nations about how many people over 60, how that proportion of people is increasing very dramatically. And with it comes the desire to live longer better. So as the organs and various bits of the body start to break down as we get older, people are looking for more and more solutions. And particularly, how can we think about bringing in engineering to help us with repairing the body? So this is now a very active field. And this sort of area of thinking about materials and engineering materials to repair the body is called tissue engineering. And this shows the general scheme of it. And it can be used in many different forms, actually. But one of the ideas might be that you would take some of the cells from the patient, so for example, stem cells. You could culture those up, and you could put them onto one of these material scaffold structures with different types of chemistry in there to stimulate your cells, and then grow that up in the lab and put it back into the body. You can also just use the materials directly in the body as well, and obviously there's also a lot of cell-based therapies. But looking at all these different ways of using materials within the body is now a very, very active field, very often performed within engineering departments. And it's very, very collaborative. So you have engineers at the heart of it, but you also have material scientists and chemists and cell biologists and surgeons. And certainly within my own research group, we have all of these different types of people coming together. And not just within your own research group, but also national networks, international networks. This is very, very collaborative international efforts in this area. And if you look at the different tissues in the body, and you can think of pretty much every single organ within the body and every single tissue type within the body, almost certainly there will be tissue engineers working on that and really developing new materials and engineering strategies to help in that process. And it's going to be very, very different for different tissue types because tissues repair differently. So if we think about liver, for example, and this is just a picture of Prometheus, who legend had it, would have his liver regenerated every day when an eagle would come and eat it. And we all know that liver maybe doesn't regenerate as well as in Prometheus's case, but it is a tissue that regenerates pretty well. And that's very different, for example, to your cartilage or how you might think about repairing a heart or your lungs. And so you need to think specifically about the tissues that you're targeting. And this is why it's very important as engineers to be speaking really closely with the medics and the cell biologists. And there's lots and lots of material strategies you can think of. So if we think about bone, for example, just as an example, and the sort of materials that might be developed for thinking about healing bone fractures, or if you'd had bone removed after a tumor, for example, then these are just some of the examples of different classes. And I'm not going to go through all of these, but the point is you can have materials made in different physical forms. So they might be made of fibers, a little bit like fiber meshes, or they might be porous, or they might be mineralizing, or they might be organic type of materials, or they might be combinations of the two. And there's many, many different strategies available. And we need to give the cells advice. So stem cells can obviously become many, many different things. And if you're going to think about using them in regenerative medicine, then you want to make sure that they're going to become the right things for the tissue that you're thinking about repairing. And this is why it's very important to think about how we engineer the materials. And if you think about a cell and the way a cell develops and the sort of information a cell needs to get to become a particular tissue, particularly if it's a stem cell, for example, then it needs to receive actually a lot of different information because it's going to have to go through different stages of differentiation and turning into the right tissue. And this can use a lot of complex chemistry, for example, and different complex molecules and growth factors that you might think about putting into your material. And all of those things can help inform your cell, and they can help in your better engineering process, but they can also make things difficult to translate to the clinic. And I think what's very important in this field is getting that balance right. So thinking about the complexity you want to encode into your material, but also making sure it actually makes it into patients. And so within our lab and a lot of the more translational labs, one of the things you want to do is really narrow down how can you make your material optimally functional, but not so complex that it doesn't go through to the clinic? And that's a very difficult balance to get right. So we think about that a lot when we think about designing these materials for regenerative medicine. And people come up actually with many different sources of inspiration and different ways of thinking about using the materials. One approach that people do is to actually think about the structure of the tissue they're trying to regenerate. So if you look at bone, it's a really, really beautifully organized tissue. So this just shows you a very, very simple schematic of what a long bone might look like. And if you zoom into it, even if you don't know anything about bone structure, you'll see straight away it's very organized. So it's got these round-looking systems. Those are called Haversian systems. They've got blood vessels in the middle, and they're all at the sort of organized at the micron-type level. And then if you zoom in again, this is a very sort of simplistic diagram, but the take-home message from it is that cells are coated, that's a cell, by the way, cells are coated in many different types of receptors, and those can interact very specifically with all the fibers within the matrix of the tissue. And those fibers of that tissue within bone are actually organized at the nanoscale, and in fact, in every tissue they're organized at the nanoscale. But in the case of bone, it's actually a very beautiful composite material where you have fibers of organic material called collagen, and then those are interspersed with small platelets of an inorganic material called hydroxyapatite. And it's that nanoscale organization of the bone that gives it these wonderful properties and also helps in the mineralization. So this has led to people trying to recreate this type of organization when they make artificial materials, and to make hybrid-type materials that have that nanoscale structure. And you can process these And you can make them in different forms. You can do clever things when you do the engineering so that you can make them either really soft or much, much tougher. And what you're going to see in this first video is a material that has that nanoscale interconnectivity of the hybrid material, the inorganic, the organic, but it's actually really, really squidgy. On the other hand, this next video is the same sort of thing, but we've changed the porosity, we've changed the cross-linking, and now it's very, very tough and very rigid. And so by using materials engineering, you can make 3D materials with all these different types of mechanical properties and also different types of biological signals within those materials and different types of surface functionalities. You can also change the physical structure really quite accurately now using different approaches. Traditionally, a lot of things that people have done are to make these scaffolds that are called microporous. So they've got micron-type pores in them, and they've got to be at least 100 microns in diameter so that you can get blood vessels growing through there when you grow your cells on there. But even though they're micro scale to a cell, and a cell is probably 50 micron or so, they actually, even though they're 3D, they actually look a little bit more like a 2D surface. And so what happens is that the cells will stick and they'll tend to elongate and flatten out a little bit. Whereas you might imagine that if you use a material with much smaller dimensions, but flexible dimensions, that the cells can move out the way and get blood vessels growing in, then that cell might be able to get interaction in 3 dimensions. And it actually changes shape if you look at how you put cells in these materials. And this is, again, materials engineering, control over material properties, ending up in a really visible biological response. 3D printing, obviously, is a very big area within this field, used in different sort of ways. So initially was used very much so in terms of just printing structures that looked a bit like organs. Now, a lot more effort, I would say, placed on also printing cells along with different types of materials and trying to start to recreate some of that hierarchy and complexity that you get within native materials. It's still not quite there yet, but very interesting developments within that field. Similarly interesting in different ways is this whole new area of thinking about electroceuticals. So how do we actually use materials to deliver electrical impulses within the body? And how can that be used as a new kind of medicine? Again, engineering very, very important within this area, and quite a bit of industry moving into this area as well. This is some work from our own group in collaboration with HMRI University. This is actually in brown. What you can see there is a single stem cell. So this is a single stem cell, and we've made surfaces coated in these tiny, tiny needles. And each cell can be interacted with actually hundreds of needles if you want. And those needles will spontaneously interface very closely with the cell, and you can deliver cells to the, deliver molecules to the cell at different points within the cell. You can also measure things going on within the cell and use this for both biosensing but also regenerative medicine. And what you can see just in this one example here that I'm showing is where we've delivered, used these needles to deliver molecules that can help blood vessels to grow. And we've delivered those into muscle tissue, and those bright green lines you can see are new blood vessels that then can start to form. And so you can think about using these technologies to pattern and get new tissues to grow in particular ways. Just want to make one slide, one point really about how materials could also be important in tackling antimicrobial resistance, and we're going to hear a lot more about that in Dame Sally's talk just after my talk. But the idea that you could use biomaterials to really control the way that infections, stop infections from spreading potentially, control the way that bacteria interact with surfaces, and also potentially the way that bacteria signal. So very, very important area that we're going to hear a lot more about in a minute. Finally, thinking about how we can use materials engineering for early disease detection. And this is important in so many different ways. So within the developed world, you might think, for example, cancer. So how can you detect if you have an early cancer? How can you detect if you've had a cancer removed and you want to know about its recurrence? If you can detect those sort of things much, much earlier and intervene much, much earlier in a therapy, the patient prognosis is massively improved. Alzheimer's disease, we don't yet have good tests, actually, for how we're going to know if patients are going to develop Alzheimer's disease. Infectious disease, both within the developed world, but also the developing world. So HIV, tuberculosis, leishmaniasis, all of these infectious diseases would really benefit from very, very good biosensing approaches using new types of engineered material systems. Our own group works a lot on this, and in the field in general, the problem you have, if you're looking at very early disease detection, so the first indications, for example, that you have a viral infection or your cancer is coming back, is that your signal is going to be very, very small. So it's exactly like this. It's trying to find a signal from a protein or a nucleic acid or an enzyme, whatever your biomarker is you're interested in, against a background of tons and tons of other stuff within the blood. And so it really is a needle in a haystack type approach, and you need better engineering and better material strategies to help you in getting that signal and making that signal much, much bigger so you can detect earlier. And one of the ways people have done this is using nanoparticles, and actually thinking again a little bit back in history, as we did with the nacre shell. Nanoparticles have actually been used for a really long time by humans, even though at the time they didn't even know they were using them. So this is the Lycurgus cup, for example, which is from the 4th century AD. This is in the British Museum. If you ever come to London, do come and have a look at it. It's a beautiful thing. And this actually has gold nanoparticles within the wall of the cup. And if you have a light that you shine from inside it, that's the picture on the left, it looks red. And if you shine a light at it, it looks green. And that's just because of the feature of those nanoparticles. When they're at the nanoscale, they interact with light in very interesting ways. And so people now that we can visualize nanoparticles better with microscopy, we can do better surface chemistry to make them and to functionalize them, people are now using these in different ways for looking at disease detection. So what you can see on the bottom left there are 2 solutions of gold nanoparticles that are aggregated in different ways due to a disease biomarker being present. And so this nanoscale reorganization of the particles, because of the presence of disease, results in a visible change that you can see with your naked eye. It's amazing. Similarly, on the right, you can also have nanomaterial systems like quantum dots, for example, that will change fluorescence if they are associated with a different disease biomarker. And that involves lots of engineering. This just shows you a schematic of how you might think about engineering those nanoparticles. Well, there's engineering involved in making the core, so a lot of synthesis involving chemists, of course, as well, but also engineering, thinking about how you tailor the surface of those materials so they'll interact with biology in the right way, and then putting on a very specific sensing surface that is going to detect very accurately the protein or the nucleic acid or whatever you're interested in. And those can be used for your clinical tools, so for very early disease detection, but also more fundamentally within research, so better tools for research as well. And that, again, is collaborative. It's got our engineers in there, it's got our material scientists, our chemists, biologists, probably some, maybe not surgeons, but certainly a lot of medical doctors also advising within that team. Within our own research, we have this center here called iSense, which is directed by UCL, and I'm a co-director at Imperial College London. And this is a really big endeavor to try and think about how we can use these types of nanomaterials for better early detection of infectious disease. And so we work on the advanced nanomaterials bit, but that's working very closely with groups developing new types of biomarkers, developing these within point-of-care type tests, so you could use these, for example, at the point of care in Africa or within other countries, in formats that you can then read on a mobile phone. And this is really important because this enables us then to do a geotagging to tell you about who has the disease and where and so on. And we've developed this in many different ways, but one of the things that we've done quite recently that was very exciting was to take these to Uganda. They've been tried in over 100 different patients that were survivors of Ebola. And with this sort of technology, we were able to actually map which patients had had which different strain of the virus and help with that geotagging and really understanding how the spread of that disease was going on. And this sort of technology is also now starting to be trialed with our partners within the Africa Center down in South Africa. So very exciting, and many other groups as well around the world helping in this. And again, collaboration, really key. This is not just our group, it's working alongside many other groups as well. And so finally, I just want to conclude by really pointing out or summarizing, I suppose, yet again, how materials engineering can be really key in terms of so many different aspects of impact on healthcare, whether that is the regenerative medicine side of things, or the biosensing, or better control potentially also of how infections spread. This is a really, really big opportunity, I think, for engineering to impact within health. Thank you very much.
Thank you, Molly. I want to introduce to you, and Molly will come back after this and we'll sit and be able to take some of your questions, so start getting those ready. I want to introduce to you Dame Sally Davies. She's the Chief Medical Officer for the United Kingdom. She's a professor. She's been the Chief Medical Officer in the UK since 2010. She is the first woman to hold that role, and in her capacity, she acts as the government's principal medical advisor. Previously, she helped develop the National Institute for Health Research. She served on the board of the World Health Organization Global Advisory Committee and the Caribbean Health Research Council. She's also the Chief Scientific Advisor to the Department of Health. Ladies and gentlemen, Dame Sally Davies.
Thank you very much. Thank you. So we're going to change pace a bit. I'm going to provoke you, because I'm not an engineer, I'm a doctor. And I'm bringing to you the problem. And it's a problem that could affect any of us. In fact, does affect all of us and leads to death. And I think you'd probably accept that a happy society, a well-functioning society, is based on a good health and a good economy. So imagine a world where when I had my cesarean section, got an infection and it killed me. Imagine a world where you have a hip replacement and it kills you. Indeed, I could show you the pictures of a little girl who was dying because she had a scratch inside her mouth with an infection, and then she received penicillin and miraculously lived. We're getting to that stage. Imagine a world where pork and chickens are so infected you don't dare eat them. We'd all turn vegetarian. It is dystopian, but actually, as I'm going to show you, this is beginning to happen. These drug-resistant infections are all around us. And in fact, you might say that the bugs are beginning to bite back. And this is a perfectly reasonable thing. This is natural selection, Darwinism in action. You put a drug that should kill the bacteria. This works for viruses and fungi too. I'm just talking about bacteria because it's easiest. You get a small mutation naturally that gives a selection advantage, and they amplify. I could go into all the details in the bottom of that slide about how bacteria don't just pass this selective advantage to their children, their progeny, but they also pass it sideways by passing DNA sideways, extruding DNA into the plasma that then's picked up by other ones, or indeed through little infective organisms. So they're clever, and this resistance is growing. Interestingly, Fleming, who was a fellow of our Royal Society and described penicillin, well, he found it, and it has saved many lives. When he got his Nobel Prize, said resistance would happen and people would die from it. And what you can see on the right-hand side of the slide is the close relationship between, and it's by country, the level of resistance to antibiotics of bacteria, bugs in a country, and the level of use. It's a clear, close correlation. And we know from science that actually the causation is there. So clearly, we need everyone's help to prevent infections. What's that? That's hygiene, that's sanitation. It's infection prevention and control. What about bubble technology for sterilizing things? It's vaccinations to protect people from infections. It's looking after our environmental discharges. But also, I want to highlight to you that we take infections with us all round the world. So when we travel, we pick up the infections that are common there. We pick up the resistances common in those places, and then we bring them home in our gut. And we all promote these infections, and I just quickly want to make sure we recognize in this room, though a lot of people don't, that actually, while we have got a lot of deaths through drug-resistant infections, this antibiotic resistance, more people are dying because they don't get access. I can show you some horrid data about how some of the good old-fashioned antibiotics, because of market forces, are no longer available. And I can talk to you also about how, across the world, more drugs are counterfeit in the low and middle-income countries than are good quality, again, promoting it. And we need to make sure we're getting this right so that we don't promote it. But why am I standing here with you? Why am I worrying about this and asking you, trying to provoke you to do something? And it's actually not just a problem, it's this complexity. So start with the fact that any animal or fish, if it takes an antibiotic, pees, that is urinates, more than 70%, often 90% of that antibiotic straight out into the environment. So we are getting environmental contamination. You can see some of that at the center of this slide. Did you know that across the world, in fish farming, and more fish is eaten that's farmed than other, the norm is to just tip antibiotics into the fish feed, so it just flows out into the rivers, into the seas. We are abusing our environment. What about when we make antibiotics? Well, the runoff from making them, if it's not properly handled, again puts antibiotics into the environment where our animals are, where our children might be. In fact, there are studies of school children in China who don't believe, according to their families, they've had antibiotics in the preceding 3 months, who had 19 antibiotics in their urine. It was so prevalent around them, let alone how we handle antibiotics in animals. This country has just changed its rules at the beginning of the year, so antibiotics are being phased out from growth promotion. That is not treating animals when they're sick, it's giving them antibiotics so they grow bigger and fatter. It's quite common across the world, and that promotes it. So it is an extraordinarily complex picture. And a lot of people say to me, well, that's fine, just get some new antibiotics. And when I was young and doctor, that's exactly what happened. We got some new antibiotics. But I want to show you here the discovery of antibiotics. And the important thing is we have not had it come into use new classes of antibiotics since the 1980s, the late 1980s. And that's in part because it's difficult to find them or to make them. There must be some engineering solutions there for you to help us with. But actually, it's to do with market failure and the expense. So, still on the subject of this, why would you care? Well, I think you'd care because you could get infections, your family could, your people could. But I persuaded my former Prime Minister Prime Minister Cameron to commission an economist, Jim O'Neill, to do a review of antimicrobial resistance, antibiotic resistance, from the perspective of the economy. And at the moment, one person dies every 45 seconds across the world of a drug-resistant infection, and that's far fewer than of cancer, as you can see here. It is not the full picture. We don't have the data, so here's a big issue, surveillance. How do we get better data, technology, and all of that coming in? But interestingly, and perhaps not surprisingly, it's impacting on the people we call the yuppies, the young, the old, the pregnant, the immune-compromised. That's people with cancer, with renal failure, with diabetes, people who've had transplantations. But if you go forward to 2050, if we don't do anything about it, one person will die every 3 seconds. This will become a common reason to die. And with that, we know that it's going to cost the economy a vast amount. It's going to drive more than 28 million people into poverty, and we are going to have a problem delivering on the Sustainable Development Goals. So what I did in coming here today was go to our research council and say, give me some examples to share to try and get you thinking not just about data, but about other engineering approaches. And I've got some examples to share with you that pick up on some of the issues that Molly so beautifully talked about, just to make you think about how you can help save lives, save our way of life. So on the left, you see an early warning system for infectious diseases using mobile technology. It's a multi-marker sensory platform. The middle one there is wearing organic sensors for healthcare, smart dressings for wound management. So what happens is, if it's one color, it's not infected, the wound, so you can leave the dressing on. If it's another color, it's infected, you take it off and you treat the infection. And then on the right, just remember that bugs put out smells like all organic things, like all living things. And so you can have an electronic nose that diagnoses infections much quicker, much better, potentially, than the old-fashioned bacterial cultures that we do. Very effective way of doing it. So that's diagnostics as one way forward. What about preventing them? And this Molly did talk about. You know, if you have treatments in hospital, you usually have a plastic catheter put into a vein. If old people often have to have urinary catheters. And the reason they have to be changed is infection and biofilms. Well, you can make special surfaces that reduce that make it much more difficult. You can modify silver or other metals and other ways of approaching it to put on new hip or bone surfaces to prevent infection. And here on the right is a hand-washing technology to produce nanoparticle-containing air and water filters that actively kill bacteria and viruses. So there you are, diagnostics and prevention. And I like this one of engineering for treatment. So that top one is a 3D printed cow shoe because it's printed specially to fit a cow that has an infection of the hoof. Now, not only does that allow the treatment underneath it and protect that cow from damaging the foot further and changing color depending on where the infection is, but it prevents that infection spreading to the rest of the herd. So instead of having to treat other cows in the hope they don't catch it, you can do it this way. And then at the bottom are some prototype bandages to treat infection where they have nanoparticles embedded in electrospun polyurethane and alginate or peptide hydrogels. And that, again, is a good way of preventing or treating infections. And the final example I thought I'd give you is engineering and public engagement. We were hearing about wonderful things earlier on. Well, games can teach people, and here's one from Bristol, an AMR comic, and Southampton having one. So this is a complex problem. And what I wanted to ask you to do now is all of you to stand up. So could you all stand up? We're going to find out who's gone to sleep while I talk. Right, now, those of you who have not had a bad sore throat, a bad infection of the throat that lasted for 2 days, sit down. In the last year, sit down. Right, so that's about right. The rest of you have had nasty infections. Now, think about how you looked after that nasty throat infection. Sit down if you did not take antibiotics, if you treated it yourself. Sit down. You've cheated. But well done, the honest ones, because in Britain, only 1 in 4 sore throats need antibiotics. Thank you, do sit down. What you've shown is how difficult it is to get this right, to actually use antibiotics sensibly so we're not promoting resistance. Think back to that picture I showed you of handing resistance from one bacteria to another so they can move it on to a cousin, an aunt, a nephew, as well as giving it to their progeny. This matters. Think about the fact that if you pick up a resistant organism in your gut when you're traveling or when you're at home, or you take antibiotics and you develop one, that it can sit in your gut, the microbiome, for 6 months, a year. And it's usually those bugs that infect people when they get urinary tract infections, or if you develop renal failure and get a bloodstream infection. They come from us inside us, yet we've already picked up or developed resistance. We have to do something about this. I've shown you the death rate, and it's very important we do. We don't know how big the problem is. We need to collect surveillance data. We need to think about mobile telephony and ways of analyzing that data, big data, to really begin to grapple with this problem, to prevent it, to diagnose it, to treat it. And there is a massive role for all bits of engineering in this problem, and I've listed some of them there, but big data, machine learning, clearly are not on that list, and I should have thought of them when I was making the slides. This is an intersectoral problem. It needs all communities working together. As Molly said, with these really wicked problems, only by collaboration will we get it right. But there's also, as you saw, a behavioral problem. Who takes antibiotics when they should, and who takes them when they shouldn't, and the interactions. So we need behavioral science. How can you design solutions that nudge people into doing the right thing? How can you help me get better data to show the politicians so they vote the money so that we have solutions to protect you, your families, your communities, our countries? We need all of these different approaches. And I leave it with you, but I would remind you this is a global problem. Infection and resistance knows no border. I spent the morning at the Department of Health and Human Services here. We are all across the world united in fighting infections, whether it's in the form of pandemic flu or bacteria. And you know, when we had a pandemic of flu across the world, the swine flu in 2009, we were worried about swine flu. We were also worried about the resistance levels of the flu viruses to the treatments we had. It's not just bacteria, it is all infectious agents, and we are all at risk. Our future, happiness and health, is in the hands of your generation, the young generation, and what you can do. Thank you.
Thank you. Why don't you take a seat here? And Molly, you take a seat here. Thank you all. Please, if you've got questions, we got a catch box out there somewhere, 2 of them over there. We've got our 2 mics over here, and I've got a bunch on the app already. My wife would have loved that, because I think of antibiotics as vitamins. Like, you know, anytime anything goes wrong, yeah. And that's what she says. She says this is what's going to happen. And now she has like a really smart person who just said that, which is how the way my marriage works. You got these forms at lunch. Just a little housekeeping. Does everybody, you remember getting these at your table? They've got a couple questions. They're going to collect them at lunch tomorrow, so I have been asked to ask you to please fill these out. Any takers for the box? 'Cause I got questions on the app. You got somebody over there? All right, go ahead.
Yeah, I guess for me, for someone who doesn't really know too much about the medical industry, and it could be a cop-out for a lot of different engineers.
I'm just going to ask you to stop for a second. I don't see where the question's coming from.
Sorry.
Oh, there you go. Okay,
great.
good. Thank you.
Yes.
For drug resistances, yes, it's a complex problem. We need to kind of traditionally approach it as we are continually making better medicines to kind of race the infectious bodies, as you mentioned. But is there a feasible solution as far as like a reset or something where we had to fundamentally change the way we fight infections, both behaviorally and in the engineering of both biomaterials and actually drugs, that could kind of start us over in the race, per se? Or is there kind of just the way we got to kind of fight the war we've been fighting till the bitter end?
Wouldn't it be lovely if there was a reset? But it's the right question. It's so complex, we can reset bits of it. We can start to make new drugs if we can find the economic incentives, but we probably need new ways of making them. So genomics comes in, but actually computer modeling with the bugs. Some of the most interesting new ones come from different ways of culturing soil to try and find new antibiotics. I think engineering has a big role to play in making sure we actually know what's going on, both at the community level, surveillance, but for individual patients, and can interrupt the development of the infections and diagnose them quickly. But it's going to be a lot of different reset buttons, not one.
Maybe okay. I can add something to that as well. So I think something that's very interesting, if you take, for example, a hip implant operation where you've put in a titanium or other kind of metal replacement, often when you put those implants in, at the time you implant them, they are sterile and they do not have infection on them. But later, down the road, there becomes an infection and you get a biofilm formation, and it's a very difficult surgery, actually, to then remove those and get rid of the infection. And so one of the ways that engineering can really play a role is in the development, and people are working on this, development of surfaces that just stop those bacteria from binding, stop them from multiplying, stop them from then communicating with each other and spreading and forming this biofilm, which is then very difficult to treat. And in many ways, if you can develop a materials approach that stops them from multiplying and from signaling, that then should not lead to resistance in the same way that bacteria get resistant to drugs. So there's many different approaches, I think, going on, but it needs a lot more work. I mean, I really liked your talk, actually, because I think this is hugely important, a massive
and not fully
problem.
Known to the degree that you know it. I mean, in your world, it may be very well known, and I appreciate that both of you have presented this in a way that engineers and engineering students can understand it and see if they can benefit from it. Let's go over there.
I have a question for Molly, and you presented a lot about technology, and some of them is already available, but many of them has been in lab for many, many years. So my question is, how far away they are going to be commercially available as a medical product to the patient they need? For example, if you want a cell phone, you spend a couple $100 to buy it. How far away from those products to be available?
It really depends on the technology and the particular problem you're looking at. So if you think about the regenerative medicine, and if you think of bone as an example, then quite a number of those technologies that I showed are actually now available in the clinic. There's other situations, for example, certain diseases of the brain and things like that, where you're much further actually from having some of those material strategies more widely available within the clinic. So it depends on the clinical problem that you're trying to solve, and it also depends a lot on the regulatory environment. So whether there's good predicate materials out there, which makes the regulatory approval a lot easier, or whether there's not, in which case it's going to be much harder. But also whether your product can be regarded as in a sense, just a device providing a physical structure, or whether it's seen as more biological. And that has a huge impact in terms of the sorts of clinical studies you need to do and the cost of getting those things approved.
I want to ask a question that's a little more general in nature, but if you were here this morning, there were definitely some people expressed concern about women in leadership roles and on the stage and around here. There's a question that's come in on the app about the types of roles you both have, not about women, but it's about what advice would you give someone who wants to hold a scientific policy advisory role? What do you think?
So not for women, but in general.
No, in general.
But
if you'd like to expand that for women, that's okay too.
Yeah. Well, I think Dame Sally has much more policy roles than I do, but I do a few things. So I'm on the board of directors, for example, of the Materials Research Society, and also the World Economic Forum Council for the Future of Materials. So and also was involved in the Royal Society's review of our research council. So I've done a few of those things, but sort of on the side. My day-to-day job is very much a professor at the university doing research, and teaching actually as well. But those opportunities are, to some extent, opportunities that have come to me. So I've been asked to do them, but they're things that I was very interested in, and I think they're very different to the day job. And so if you can be involved in things that really enable you to think in a different way and to have impact on a larger scale, I think it's really interesting. But those opportunities tend to come as you get a bit more senior, and I'm sure Sally will have a lot to add.
You're in the ultimate policy advisory role.
It's great fun being an independent advisor to government. So I think there are two routes. You can opt out post-PhD or postdoc into generic policy, whether it's health or science, but then you'll be moved around areas, and some people really enjoy that and develop great careers. Others do it, as Molly and I have both done it, based on our own scientific expertise, starting with committees and working on those, and then showing that we think not just only about the science, but the role of science in society, because policy is, for me, it's about the role of science in society. How can science assist our society? How should society interact with it? So if you get these opportunities, take them, show you're good, and then there are always more people who will ask you to join in and do more. And that was how I did it. I spread slowly across and eventually held my nose and jumped, and luckily it worked.
Fair enough. All right, thank you. Over there, sir.
So, yeah, I'm just wondering, like, the use of antibiotics for livestock and also people misusing it, for instance, doctors prescribing it when it's not needed. What can we do as a group, but also influence government policies to make it so that this isn't a concern, as it's largely contributing to antibiotic resistance?
So there are two routes in. How do we help the doctor-patient relationship? if we had really good rapid diagnostics, so a doctor didn't say, well, you've got a sore throat and I don't think it needs antibiotics, so please go away, but we could actually, within only a couple of minutes, say, you don't need them or you do. You can integrate into the IT systems some kind of artificial intelligence, and I'm sure over the years that'll improve so that doctors get not only quizzed, but pushed in certain ways to get the right things. And you can start to raise awareness and start to change how we regulate the whole system, which is more the political side. I think we are raising awareness about it, but we keep ending up talking about doomsday. And at the moment, it's only one person every 45 seconds across the world who dies of a drug-resistant infection. I mean, my golly, that's nothing, isn't it?
But I'm still wondering, when it comes to livestock and farmers using it because it's an easy way of making the animals grow quicker and resolve the solution, how can we, put policies to make it so that this isn't done as much?
Well, in Europe, we have not, since I think 2003, allowed antibiotics for growth promotion. So we are having a great move through the United Nations. I chair their Committee on Antimicrobial Resistance, which is, we call it, One Health. It brings together the animal sector of the UN, the human sector, and everything, and we're trying to get all the countries to agree to change their regulations. That needs the public. It needs new ways of looking after animal husbandry so that in poor communities they don't see it as the only way they can protect their animals from infection. So we need better sanitation, better infection prevention and control. We need new ways of giving growth promotion, whether it's prebiotics, probiotics, or other things. So that, again, as a previous questioner said, there isn't a single magic bullet for any of this, which makes it great fun, but horribly difficult.
Is it?
Okay, I got to move on. Got a lot of questions. Thank you. Go ahead.
First, I want to say thank you for your time for answering these questions. And I wanted to start saying that I'm from a third world country, and we don't have access to a lot of medications over there. And now it comes to my mind that when I had an infection or a wound, my mom and grandma would use plants that are popularly, known within the population to work, and they actually cure the infection. So I'm now thinking, also in the example of a doctor in Congo who saved 18 people from malaria using a plant by pulverizing the leaves and applying it to the wound. So I'm thinking also that bacteria have been around for millions of years, and plants and animals interact with them all the time. And over those years, they developed the tools to fight resistance. And as an engineer, don't get me wrong, I love to create new things, but maybe we're not thinking on this medicines that nature has already created for us, and we need to basically just look for them in the right places. So maybe we could skip this creating process and actually get better at identifying plants that actually work at the moment. Because I feel like since science was discovered...
That is a wonderful point. If you look at the best anti-malarial, it's artemisinin, and it is from a traditional Chinese medicine. If you look at our best antibiotics, they've come from bugs and fungi in the soil. Germany has a very exciting program of looking at insects and finding natural antibiotics from them. So where do you engineers come in? Well, it's one thing finding it, but then how do you make enough of that? What are the fermentation or growth or synthesizing methodologies to bring it to scale once you've found one? And only last week, we launched a bilateral fund with China, 10 million from each country, and one of the objectives is to look at their traditional Chinese medicines for new antibacterials. So you're right, and we need to do more.
Thank you.
Thank you. And listen, this has got to be one of the smartest things I participate in any year, but we do get into smart people-itis, where the questions become theses. So I do want to have questions, please, and just ask the question, and I want to get through with a lot of people. Thank you.
All right, so on the question, I remember visiting India with family and having a sore throat a couple years ago and being really surprised at how easy it was to get antibiotics. They seem to be kind of given out at a drop of a hat. And like you said at the end of your talk, bacteria, viruses don't really know borders. So what role do each of you think that these large nationally associated bodies like the National Academy of Engineering, the Royal Academy, and the counterpart in China have to play on kind of the advocacy and the policy front, along with the engineering work that we're all doing at the same time?
Well, I don't know that you have a direct role as engineers in advocacy. You do in solving the problems. But where our Royal Academy plays in is with the Royal Society and the work on advocacy the whole scientific academy is doing about this issue. So please join with your colleagues and do it together.
We got a catch box out there? Yeah, somebody's got it. Can't see you, so there we go. Got it. Thank you.
How well characterized and classified are these resistant antibiotics, or the resistant bacteria? And how good are we at this point at pinpointing their location and saying, oh, this drug didn't work because there are resistant, the resistant bacteria?
So there's a lot of whole genome work being done on bacteria, and with really good specificity, they can now say this mutation means resistance to that antibiotic because of this pathway. So there's really increasing characterization. What we need is point-of-care genomics. And actually, we've got variants of that in the HIV clinics looking for resistant HIV and TB now in Britain, and it's spreading elsewhere. We diagnose TB directly rather than from cultures and do the whole genome and say, this one's got resistance, you need that regime, that one hasn't, use the ordinary regime.
I can maybe add also some sort of unpublished work that's not out there yet, but there's quite a bit of effort looking at how you can use new types of chemical mapping and chemical imaging actually within the wound bed. So to try and identify within a wound bed where you have the bacteria, but also exactly what type of bacteria it is. And I think that will be transformative, because if you can be within the clinic and have your probe that then does a chemical fingerprint and tells you, is that one drug resistant, is that one not, so that you know how to treat. I mean, that's not available yet, but it's coming.
That'll be great. Yeah, fabulous.
I have a question on the app directed to both of you. Both of you are excellent communicators of complex STEM concepts and have proven track records of funding and support. What advice do you have for young engineers who are still learning the art of communication and how to engage with the public?
So, you can start if you want.
Well,
see, they're polite. That's a good start. You go first. No, you, please.
I have to communicate to a variety of audiences. And I think the answer is I use a different style for different audiences. So if I'm doing TV, it's like talking to one of my patients, of which I've had years of practice, which is going back to first principles using simple language and stories. If I'm talking to politicians, it's quite a different issue. I put myself in their position and think about what would a voter say and what would the newspaper say, and I talk about it from that perspective. If I'm talking to scientists, I try and talk about the scientific issue. So the answer is, I have learnt that different people hear in different ways, and I moderate what I say for the audience.
I mean, I suppose I give an awful lot of talks, actually, probably at least one a week or so, but most of them would be very, very technical. So at very specialist scientific meetings, and my slides would be full of a million graphs and lots of data and things like that. And so these kind of talks, actually, I would give less often, maybe on these more general type talks, maybe 2 or 3 a year, something like that. And it is more about getting general concepts across rather than very specific pieces of data. And then obviously those of you that are very interested in the specific science behind it at least know what things are around, and then to be able to delve deeper into it. But
Ali, what works?
Yes, actually, you're probably the
I think the point you made about knowing reading the room, knowing the audience. We have had some conversations today which have been way over my head. And I mean that with great praise, because the panelists knew it, and the questioners knew what you were talking about, and the connection was made. And there have been some conversations that have been much more general, and again, the panelists know what they're being asked, and you know what you're asking. So finding the place at which you're meeting with the audience, if it is the public, change your message so that the public can hear it, and if it's a specialist, adapt your message as such. This question has come up in different forms a few times today, and I'm glad you're asking it, because it is something that I know the folks at the National Academy of Engineering struggle with, and that is, what's the messaging behind the importance of engineering and the work that engineers do, so that they are well regarded and they are at the policy table as decisions are being made. So really good question. Thank you for asking.
There's another side to that, which I find very useful. Sometimes it's much better if I don't give the message, but I get the engineers to give the message, and sometimes it's better for the engineers if I give their message. So getting other people to give the message too.
Very good. All right, excellent. Sir.
I'm interested in seeing your view on the use of antibiotic rotation in solving drug resistance.
You're getting into real specialist stuff. So the concept is that if you stop using an antibiotic, the resistance levels will drop. And there is some evidence, which you clearly know, that it does drop to undetectable. What I don't think the literature tells me well enough is how quickly it returns when you reintroduce the drugs. But it clearly, there are stories of clinics where it has worked, where every 3 months they cycle the drugs, and we don't do that in Britain yet, and I've got colleagues who are doing experiments on that.
We're at a university campus, so there are all these things where hand sanitizers all over the place, and we have a question on antibacterial hand soaps. Are any of these related to this conversation or no?
Yes, and it's part of infection prevention and control.
So you like them?
No, I like alcohol.
Okay.
And I like...
That is officially the biggest round of applause we've had this entire conference.
Right. That'll just be a soundbite now.
That's right.
For the non-British, there are Chief Medical Officer guidelines of low-risk alcohol drinking, which make, and I've been done over by the newspapers repeatedly on them, but it's evidence-based. God. And I like soap and water, but I'm told there's work coming which suggests that some of these other antibacterials may drive some resistance.
Interesting. Okay. So kids, maybe, because when I was growing up, we were just left to be a little dirtier.
Yeah, not a bad thing.
So yeah, there you go.
I think that's not a bad thing either, as a kid.
Yeah.
Yep.
Hi. So I really enjoyed the conversation this morning about women in engineering, but I was just interested to see what your experiences of that, and I suppose sexism in general, and then what advice you'd give to a male growing up in that kind of situation, and what we should be doing
to protect your future or make the situation better? You have to finish the question. That's okay. We'll finish it for you.
So we have a few different things in the UK now, and I'm sure it's the same in the US and other places, around trying to address some of these issues and some of the things you might do. And this is called the Athena Swan program. And I think one thing that's important in that is that actually if you look at it as just something specific that you're fixing for women, I don't think that's quite as effective. But if you can look at it as how can you make the environment better, for women, but so that it's better for everyone, then that is going to have a lot more carry through and be a lot more effective. So a lot of our Athena Swan practices that we do, they turn out being really good for guys as well that have, for example, families, but also they want to be more inclusive in general and just have a better working environment. And obviously a lot of the male professors will have a lot of females in their group, and the better you make that environment, everybody benefits, right? Not just the women, the men, the science, everything gets better for everybody. So we have programs in place for that. I've seen definitely some really good improvements within the UK departments with having those sort of targets in place, but there's obviously a lot more that can be done. Yeah.
So that's, Athena Swan is a charter mark. And I set up the National Institute for Health Research in the UK and persuaded the government to give me £1 billion a year to spend on clinical research. And about 4 years in, I thought the women were getting a bad deal. So I actually sent out a letter to all universities, medical schools, hospitals, saying, next time you come for these grants, which were worth £850 million, you will only be shortlisted if you have the Athena Swan Charter Mark at Silver Award. I have never had such an unpleasant, it was even worse than the alcohol, unpleasant post bag from heads of universities. But it absolutely changed things, because they did. I mean, I'm tough as old boots. They did have to get the charter mark, and it changed things. I think we now need something else. And I wanted, as Molly said, to change it for women, but actually for all people. I thought that if we got it right for women, it would work for everyone. But the post bag was not nice.
All right, well, it's good to know, because you're both so well positioned, as others have pointed out, and as we know from the histories of the positions you both hold. It's not that there have been women all over the place doing this. This has been hard. You have both, it's hard won, and that battle is not anywhere close to being over. Please.
Hi, there's been a lot of information recently, both in terms of drug-resistant strains of bacteria and also vaccinations and other health issues out in the world that affect really everyone. And while engineers and scientists maybe have an easier time understanding that information, the general public doesn't have as easy a time digesting that information. Are there tools that we as engineers and scientists can recommend to people who don't have scientific backgrounds to help them consume information and actually help with the efforts to control things like the overuse of antibiotics, tools for that?
Yeah, I mean, I must say I have heard what you said today was the first time that registered as fully with me as it did. I don't know that I've seen that before in that way.
So I've done a TED Talk, I've written a book. There are comic books for children. We are working with the Wellcome Trust to try and get it into school children's education packages. It is a slow grind. There's quite a bit of stuff on the web. And in fact, Public Health England, which is our public health agency, has a program called eBugs that's now in more than 20 languages and used in lots of schools. So we do try. We've also been piloting, and I'm waiting when I get back to see the impact of it, a TV advert in the northwest of Britain. And it's very interesting when they did the basic research, they call it, but it's market research, into how people perceived this issue of doctors saying, no, you don't need an antibiotic. They thought the doctors were trying to save money. So it's Saatchi and Saatchi actually have done this amazing thing of dancing antibiotics, saying, look after us or we won't be there when you need us, which is very clever, a totally memorable jingle. But did it work? I'll find out next week.
I think another thing to add is, I mean, it's interesting actually your comment saying you pop them like vitamin pills, because for me, you know, I just wouldn't even dream of doing that. And
my wife doesn't let me. I just want to.
Yeah. Oh, you just want to. But part of the reason is that actually, of course, this antimicrobial resistance is a major problem. But the way we would see it now is, okay, it's a major problem coming. It's maybe not quite as tangible, and you think it might happen to someone else and not to you, and so on. But if you look at all how medical, a lot of new things are coming into medical science now. One of the things that I found so fascinating is the role of our microbiome, so all of your natural bacteria within the body, and how that can help prevent you from pretty much every disease, actually. And every time you take an antibiotic unnecessarily, you're knocking that out. And so it's not like you're just being neutral by taking it. You're potentially being detrimental to yourself even right now. So
it may be a generational thing, though. When you showed that chart of development of antibiotics, you know, I'm at the end of a generation that thought that antibiotics make us live longer, right? We have our longevity. Otherwise, we'd all be dead if not for
and
antibiotics.
And in Africa, that's still true.
In my mind, antibiotics save your life,
well,
so why not take one?
They do if you have an infection, for sure.
Yeah.
Yes.
Very interesting.
But if you don't, you're better not taking it.
But picking up on Molly's point, it begins to look as if taking lots of antibiotics, particularly in children, predisposes to obesity because of altering the microbiome. And in older life, probably there's more diabetes because of taking it. So we're beginning to understand that not only do you get resistance, but other things increase your risk of other illnesses.
And I don't think that message is out there yet, right? About the microbiome, because this is all science that's really evolving. Like at the moment, if you look at journals, you know, even last week, there'll be new papers on this. So I mean,
once in a while it makes it into the mass media because people talk about a fecal transplant or something of that nature. So it's got a headline or something like that, but it's not something everybody talks about. Sure.
I believe, I'm not 100% sure if this is correct, but is it true that you can combine antibiotics for treatment, try and combat infection with multiple antibiotics?
Absolutely. So we now know that for effective cancer treatment, in general, you want combination treatment. For effective TB treatment and HIV treatment, they're combinations, and malarial. So why on earth do we not do it for bacterial? I keep asking the experts in this field, and they say, yeah, we just never have done. So we'll probably end up there.
Okay, and how much time would that buy us, do you think, before we have to find some alternative solution?
Well, we do need alternative solutions. So I'm working through our government with the G20, group of 20 nations, to try and see if we can find better economic incentives to pull the investment into better basics or more basic science and better development of drugs. We need a lot of work.
Okay. All right.
Thank you.
We have a catch box? All right. I feel like the catch box is staying in the same place and people are just rotating, but it doesn't matter. New questions anyway, so go ahead.
Hi. I know we've talked a lot about prevention and, you know, how to prevent, you know, antimicrobial resistance and evolving it. But do you ever foresee a way for us to predict the next mutation, to use all of the data from all the microbial resistance that we have gathered so far and to track how certain microbes, certain bacteria have actually mutated and to predict their next mutation?
I think some of our genomic scientists with bioinformaticians are already using big data to see where are the more fragile bits of DNA and why and where you might get mutations. So I think that is not sci-fi. I think it's the stuff of the future that's beginning to come. And you can see also, I mean, the problem with this transfer laterally of resistance is you can take resistance from one type of bug, an E. coli, and it can transfer to Klebsiella too, so that you get that as well. It's not just in one type of bug.
Sir.
I just want to thank you both for traveling to the United States today for this conference and ask, you mentioned the right data and helping you convince politicians. So what are ways engineers can provide solutions to help bring down the cost of prescription drugs?
Well, I think that we have standard ways of producing drugs. And the West, the pharma model of the West is based on that, whether it's drugs or vaccines. And then when you look at what the Indians do, they use process engineering and innovation to absolutely change how they're done. And so I think we need to get more brains that are good at process engineering and looking at the real chemical processes and saying, just because you always did it that way doesn't mean it's the right way. There are better ways. And I think the Indians have led the way on drug production and vaccine production.
That's a good question. Thank you.
So in my experience, biomaterials research and also antimicrobial resistance research is all very high level and more in labs and expensive. So what is the timeline that we can think of for these to become more low cost and available to very developing countries and impoverished areas of the world?
I think there's just very, very different levels and grades of that research. So, I mean, if you look at some of the simpler technologies that you could manufacture more readily, sterilize easily, etc. I mean, lower hanging fruit type things, whether it's better bandages or whether it's sort of off the shelf implants that function as a more physical role. Those things are actually relatively faster and easier to translate. If you're looking at something in terms of a biomaterial where you really want to try and recapitulate a lot of the growth factors and things like that involved in development, then that's not only very, very complex, it's also horrendously expensive and has a very high regulatory cost in terms of the time involved in that. So it really depends on the complexity of the intervention that you're going with. But I mean, certainly within the UK now, we have a number of different funding streams that are around funding technologies for use within a more global setting, and we're involved in a lot of that work as well, where, for example, we're making minimally invasive implants that you could use for much quicker, faster, easier hip surgeries and things like that within a global health settings.
Yeah. Thank you.
All right, well, that is the end of this session, but what a terrific discussion. Thank you for bringing us your knowledge, for sharing it, and for getting everyone involved. I think you should do everything you can to follow Molly Stevens and Sally Davies and the work that they're thinking about and that they're working on. I'm going to hand this over to someone else now for a very special session that you've got coming up. You're going to hear from student speakers from the Grand Challenge Scholars Program. I'll be back with you all day tomorrow, starting bright and early. You are about as great a crowd as there is because you're all punctual and you come, you do your homework, you've got great questions. So I want to thank you for a very productive day from my perspective and for only making me seem stupid for about 45 minutes. Thank you, everybody. Stay where you are. We got another great session coming up.
Thank you.